嘉宾，大家好，欢迎各位先进参与2022年第3届台湾国际微肢体研讨会。我是工研院生医所郑淑珍，也是此次研讨会的主办人。本次会议是由日隆精化国际股份有限公司及日本精化株式会社主办，由环球生技协办。今年台湾国际微肢体研讨会的主题是微肢体技术在奈米药物与药物传输的新应用。我们邀请了多位台湾跟日本在微肢体领域的专家学者，用影片预录的方式来与大家分享微肢体新型开发的技术跟应用发展的实况。在研讨会中，我们也会透过语谈的分式来分享微肢体的新应用与展望。在会议开始之前，让我们首先邀请日隆精化陈黄总经理来为大会致辞。
日本精化株式会社实野社长，德岛大学石田教授，国立台湾大学陈进庭教授，以及工研院生医所，标靶药物与传输技术组郑淑珍博士、郑平福博士，还有各位与会嘉宾们，大家午安。首先代表日隆精化国际股份有限公司，欢迎各位产学先进共襄盛举，谢谢大家的支持，让台湾国际微肢体研讨会迈入第3届的国际交流。日本台湾学研及产业界在质质科学方面，不仅拥有丰富的开发经验，在国际医药领域更拥有卓越贡献，不断吸引来自产官学研医界等不同领域的专家投入。而微肢体技术的研究运用至今已超过1甲子的发展，从全球第1项微肢体药物英国的安必耸获到批准，至今也有非常广泛的发展。微肢体作为药物的载体，透过提高药物稳定性、降低药物毒性，达到缓释或标靶的功效，能够充分解决给药上的许多难题，因而被认为是迄今最成功的药物载体系统。我们也看见许多被全世界广泛使用的产品，都已经和微肢体技术互相应用，进一步成为药物的最佳载体。而随着全球生物技术的急速进展，我们也观察到了微肢体在新领域的突破。除了过去最多应用的肿瘤药物之外，2018年第1项 siRNA 药物 Alnylam 的 Onpratto 得到美国 FDA 批准，而风靡全球的 mRNA 疫苗也透过磷脂质的包覆，有效保护序列被裂解，利用其靶向性协助辨认，进而增强转译过程及效率。这些后疫情时代的崭新应用，也将带动微肢体开启核酸药物发展的新纪元。当 mRNA 与微肢体的搭载技术日渐成熟，我们可以预见研究落地的情境将成为众所瞩目的未来发展新契机。因此，今年度的主题定为微肢体技术在奈米药物与药物传输的新应用。我们非常荣幸能够持续邀请到台湾微肢体领域重磅专家共同与会，并与日本跨国连线，分享微肢体领域最新的技术进展。希望透过聚焦新药开发、再生医疗以及核酸产业，学者先进们能够互相交流，带动台日生技产业的合作发展，促进国际微肢体产业链的建构，建立专业人才的培训机会。相信这些都是需要依靠大家齐心协力，共同努力来完成。最后，我也敬祝各位与会嘉宾身体健康，研讨会圆满成功。谢谢大家。
谢谢陈总经理的致辞。接下来我们邀请日本精化株式会社石野浩夫社长来为大会致辞，并且介绍日本精化株式会社。
代表取締役社長、また日重製菓の当時長を務めております矢野博と申します。どうぞよろしくお願いいたします。この度第3回台湾リポソームセミナーを本日オンサイトオンライン共催という形で開催することになりました。まずは会社を代表いたしまして本日のセミナーに出席していただいた台湾の皆様、日本の皆様、感謝申し上げます。また本日講演していただける鄭先生、陳先生、日本からは石田先生、ありがとうございます。重ねてお礼申し上げます。セミナーに先立ちまして、まずは当社のご紹介をさせていただきます。日本製菓は国内商農事業の統一を目的に、1918年に日本商農株式会社として設立されました。その後、脂肪酸やその他油脂関連製品の製品販売を開始し、独自の科学分野を切り開き、業用を拡大し、1971年に現在の社名、日本製菓株式会社と改めました。以後は、科学を通じて社会に貢献することを経営理念とし、現在では化粧品原料、衛生管理製品、医薬品原料、電子材料等を製造販売するファインケミカルメーカーとして時代のニーズを先取りする製品開発、事業展開に取り組んでおります。本日ご紹介する当社のリピット事業は1991年から医薬品向けに商業生産を開始し、その後多くのリポソーム医薬品に採用され、今も製造販売を継続しています。当社のリピット事業では、独自の生産プロセスにより、医薬品原料用の高純度リン脂質、高純度コレステロールを生産しております。これらの製品はいずれも CGMP の管理下で生産しており、米国当局である FDA をはじめ、欧州や日本等の当局により、査察実績も豊富です。また、最近では、米国製薬企業であるギリアード・サイエンシズ社が製造販売するリポソーム製剤アンビソームに関するアライアンスにより、新工場建設と昨今のアンメットメディカルニーズへの対応や、拡散医薬品などの新しい医療モダリティへの多様化への対応を目的とし、総額58億円をリピット事業に投資いたしました。現在、当社の高砂工場内に2つの新しいリピット事業専用プラントを建設中で、2023年より順次稼働する予定です。さて、我々のリピット事業は2018年に台湾市場に参入いたしました。2019年10月に台湾の台北市で第1回目のセミナーを、昨年2021年10月にはオンラインで第2回目のセミナーを開催いたしました。いずれも多くの大学や企業から参加していただきました。台湾の企業様からは、リン脂質やリポソーム等の問い合わせも多くいただくことができ、セミナー開催の影響力を感じると同時に、台湾進出の大きなきっかけになったと確信しております。 2020年初頭より世界中で蔓延している新型コロナウイルス感染症は、我々の生活や価値観を根本から覆し、環境を一変させました。この影響により、様々な学術集会や研修会が中止、もしくはオンラインでの開催に迫られました。我々も2020年には本セミナーの開催を見送り、翌2021年にはオンラインでのセミナー開催を余儀なくされました。ようやく収束が見え始めた今年に、オンサイトでリポソームセミナーを開催できることを非常に嬉しく思っております。世界の医薬品業界において、新型コロナウイルス感染症のワクチンとして一躍有名となった2つのメッセージャー RNA ワクチンの登場は、業界を揺るがす地殻変動になっております。そして、リン脂質を用いたリポソームや LNP などのドラッグデリバリーシステムに、これまで以上に大きな注目が集まっております。本日ご講演いただく鄭先生、陳先生、石田先生は、これらの分野のスペシャリストでございます。先生方のご講演を拝聴できることは、私自身の大きな喜びであり、本日参加されている技術開発者の皆様にとっても貴重な時間になると確信しております。ぜひ活発に議論していただき、参加いただいた皆様全員にとって有意義な時間となることを願っております。また、本日のセミナーを通じて、私どもの事業活動についても一層のご理解をいただき、これを機にさらに信仰が深まれば幸いでございます。本日はどうぞよろしくお願いいたします。
谢谢石豪副社长对这大会的致辞，也是对台湾跟日本在微肢体上面技术上面的交流。接下来我们今天第一位邀请到的讲者是 Mr. Yuki Inoue。Yuki Inoue 毕业于京都大学化学系，目前任职于日本精化株式会社。那今天演讲的题目是要来介绍日本金氏株化会社的 phospholipid 跟他们用特殊技术所制造的 Liposome。让我们欢迎 Yuki
Hello, my name is Yuki Inoue, and I'm a researcher from Lipid Division of Nippon Fine Chemical. We call our company NFC. Today, my presentation consists of two topics. First, I would like to introduce preparation of liposome using Liposome. Liposome is a lipid mixture produced by our own technology. Liposome can significantly shorten the complex process of liposome production and is expected to contribute to SDGs because it enables to prepare liposome without the use of organic solvents. Liposome is capable of producing up to 20 kg per lot under GMP control and has been used for liposome formulation on the market. We can produce liposome with any composition you require, and we can also produce liposome containing liposoluble drug. Typical liposome preparation methods include mechanochemical Van-Gun, reversed phase evaporation, and solvent injection. These three methods use organic solvents are difficult to scale up to remove the organic solvents and have the risk of organic solvent residue in the final product. The mechanochemical method shown here generates heat during liposome preparation, which may lead to degradation of the material contained in the liposome. Each method has its advantage and disadvantage. The Van method, reversed phase evaporation method, and solvent injection method introduced earlier are used for test tube level production, while polyhydric alcohol method, mechanochemical method, and heating method are used for beaker level preparation. When scaling up for commercial production, it is often difficult to scale up using methods that we prepare at the test tube level. Reproducibility of liposome production may not be possible when changing liposome preparation method in formulation development. In addition, changes in preparation methods may limit the solvents that can be used, and changes in scale-up may result in changes in solvent removal levels, which may leave solvent residue in the product. These points are important to ensure that scale-up can be performed without problems. The first is to create a homogeneous lipid mixture at the molecular level. The second is to establish a method for removing organic solvents. The third is to have high hydration of the lipid mixture. Low hydration requires new equipment or process. Liposome meets these requirements and eliminate many of the problems associated with scale-up. Preparation of liposome using liposome is explained. MLV liposome suspension with a particle size of about 1 micrometer can be obtained by simply adding an aqueous solution to liposome without using organic solvents. The liposome suspension can be sized to the desired particle size. Liposome is not simply a mixture of lipid powder or a mixture of lipid solution from which the solvent has been removed, but a mixture of lipids at the molecular level achievement through a special process for the preparation of liposome. It is ideal for simplifying manufacturing processes, mass production, and prepare high concentration liposome suspension. We compared hydration ability of liposome and only blended lipids. The photo on the left shows the blended lipids of HSPC, MPEG 2000, DSPE, and cholesterol HP, and the right shows liposome ACD1 prepared with the same composition as the blended lipids. Aqueous solution is added to liposome and blended lipids. The solution was then hydrated by shaking them by hand in a warm bath at 65 degrees until all lumps had visibly disappeared. The shaking time was 6 minutes for both. The blended of 3 lipids had a lot of foaming, whereas the liposome had less foaming. The hydrated liposome was subjected to particle size reduction using the extruder at 65 degrees. The liposome with blended lipids passed through the extruder in 1 minute, whereas those with the liposome took about 3 minutes. The liposome after extruding is shown in the photo here, with the lipid mixture on the upper side and the Liposome method on the lower side. The liposome with Liposome is whiter than the liposome with lipid mixture. In the case of blended lipid, a large amount of solids precipitated during extrusion and were not included in the liposome, whereas in the case of Liposome, no residue was observed during extrusion. This may be due to the difference in lipid concentration in the liposome. Next, HPLC analysis was performed to examine the solid. The MPEG 2000 DSPE and HSPC peak showed no difference between the blended lipid and the Liposome, while the cholesterol peak was smaller in the blended lipid than in the Liposome. This suggests that the liposome with the blended lipids contained almost no cholesterol and that the solid were cholesterol. The reason for this is thought to be that cholesterol is highly hydrophobicity and therefore could not be incorporated into the lipid membrane by simply mixing and hydrating each lipid. As a result, the liposome composition changed from the initial ratio when blended lipids were used. On the other hand, the liposome with Liposome almost incorporated cholesterol into the lipid membrane. We measured the IR of the blended lipid and Liposome to investigate why cholesterol is incorporated into liposome in Liposome. We found the difference between the IR results of the two blended lipids and Liposome. This suggests that Liposome is not a mixture of powder with each lipid having its own crystal structure, but that the crystal bonds of each component are lost and new intermolecular bonds are formed. The Liposome was a solid that could be easily hydrated by mixing down to the molecular level, and cholesterol could be incorporated into the liposome. This is the process flowchart to manufacturing liposomal drug like Doxil using Liposome. First, you measure Liposome and buffer solution and stir. Second, sizing by using extruder. Third, dialysis from ammonium sulfate salt to sucrose. Fourth, loading API into liposome by using remote loading method. The preparated liposome are shown here. We studied characterization of liposome accordance with FDA guideline. Lipids and doxorubicin concentration were determined by using HPLC methods. These concentrations, drug-to-lipid ratio, and the percentage of encapsulated doxorubicin were almost the same with Doxil ones. We would like to introduce our manufacturing facility for the production of Liposome. The production of Liposome used in the pharmaceutical products is performed entirely in an environmentally controlled area. After dissolving raw material in a solvent, the product is processed using the crack system shown in the photo, dried in a dryer and packed. The process of manufacturing process of Liposome is FDA and EMEA approved. Liposome is a lipid mixture for liposome preparation. It's easy to produce liposome dispersion in large scale by use of Liposome. This table shows examples of Liposome products which have ever been manufactured. Liposome ACD1 is registered in the US Drug Master File. It is also available as a test reagent from TCI. The lipid composition can be freely combined according to your request. I would like to then introduce charge-reversible lipid RNA delivery applications. There are 3 nucleic acid drugs on the market that use LNPs. The ionizable lipid used in the marketed LNPs all contain tertiary amines and are positively charged under acidic condition, allowing negatively charged nucleic acid to be encapsulated. However, these ionizable lipids have their own problems. For example, the manufacturing process is complicated and PEG-modified lipids, which have been raised in the side effect, are required to form stable LNPs. We have developed a new lipid derivative, DOPDEDA, in collaboration with Professor Tomohiro Asai of the University of Shizuoka. The structure of DOPDEDA is shown here. DOPDEDA is phospholipid consisting of hydrophobic portion of diacylglycerol with a hydrophilic portion of diethylengyamin, which is pH-responsive head group. In addition, we have developed DHSM-DEDA consisting of a hydrophobic ceramide with a hydrophilic portion of diethylengyamin, which is pH-responsive head group. These lipids have charge-reversible property due to their structure, which contribute to intracellular drug delivery and low cytotoxicity. These phospholipids can be synthesized in a one-step by enzymatic reaction. Furthermore, LNP can be prepared without using pegylated lipids and without aggregation. The following is detailed description of its structural characteristics. The diethylengyamin group in the phospholipid head has the characteristic of being positively charged under weakly acidic condition, neutral under physiological condition, and negatively charged under weakly basic condition. This makes it possible to obtain low cytotoxicity, high nucleic acid encapsulation effect, high cell permeability. The structure of LNPs and liposome, a well-known DDS technology, will be compared. LNP is monolayer and their interior is packed with lipid, whereas liposomes form a bilayer membrane and have an aqueous phase inside. The preparation of LNP is simple. An alcohol solution of lipids and an aqueous solution of nucleic acid are mixed in a microfluidic chip, and the alcohol is removed by dialysis. The physical properties of LNP are shown in the table here. The particle size about 100 nanometer and the nucleic acid encapsulation rate can be prepared at about 100%. We introduced the pH response of LNP prepared with DOPDEDA or DHSM-DEDA. LNP with DOPDEDA or DHSM-DEDA were found to be negatively charged under neutral condition and positively charged under weakly acidic condition. The number of dead cells was evaluated by LDH assay. The results showed that these LNP were comparable to the negative control indicated low toxicity. It was also confirmed that these LNPs were significantly less toxic than those using cationic lipid made by other companies. These results suggest that LNP using DOP-DEDA or DHSM-DEDA can induce gene silencing without damaging the cell membrane. The hemolytic property of LNP was then evaluated by hemolysis assay. It was found that hemolysis was observed under weakly acidic condition, pH 5.5, where DOP-DEDA or DHSM-DEDA is positively charged, while under physiological condition, pH 7.4, little hemolysis was observed. These results suggest that these LNP do not affect the membrane barrier capacity under physiological condition, but destabilize the membrane in weakly acidic endosome and promote endosomal escape. GFP assay was used to evaluate the effect of the target gene silencing in vitro. The results suggested that LNP with DOP-DEDA or DHSM-DEDA may have the same level of target gene silencing effect as cationic lipid produced by other company. The organ selectivity of LNP was confirmed using mice. The results are shown in the image. It is clear that LNP accumulate in the liver, which is target site, while naked siRNA accumulate in the kidney and other organs, in addition to the liver. We would like to introduce DHSM-PPZ, a derivative of lipid DHSM that improves blood retention. The half-life of liposome containing DHSM is significantly longer than the liposome without DHSM. Furthermore, when compared to liposome containing PEG, a lipid that contributes to blood retention, it is also been confirmed that the half-life is comparable at the right concentration shown on the right. As a comparison, we have also developed DOP-PPZ, a phospholipid consisting of a hydrophobic portion of a diacylglycerol. They are similar to DOP-DEDA and DHSM-DEDA and are amphiphilic and pH responsive. Therefore, intracellular drug delivery and low cytotoxicity may be achievable. LNP are prepared by mixing an alcohol solution of lipids and an aqueous solution of nucleic acids in microfluidic chip and removing the alcohol by dialysis as described earlier. The physical properties of the prepared LNP are shown in the table here. The particle size is about 150 nanometer and the nucleic acid encapsulation rate for DOP-PPZ is 94%. For DHSM-PPZ, the optimal conditions are still being investigated and are currently around 67%. Regarding the pH response of LNP with DOP-PPZ or DHSM-PPZ, it was confirmed that they are negatively charged in neutral condition and positively charged in weakly acid condition. The results of cytotoxicity evaluation were also confirmed in the same experimental system as DOP-DEDA and DHSM-DEDA, showing that LNP is as low in toxicity as the negative control. It was also confirmed that LNP was significantly less toxicity than cationic lipids made by other companies. These results suggest that LNP with DOPPPZ or DHSMPPZ can also induce gene silencing without damaging the cell membrane. The hemolytic property of LNP was then evaluated by hemolysis assay. It was found that hemolysis was observed under weakly acid condition pH 5.5, where DOPPPZ or DHSMPPZ is positively charged, while under physiological condition pH 7.4, little hemolysis was observed. These results suggest that these LNP do not affect the membrane barrier capacity under physiological condition, but destabilized the membrane in weakly acidic endosome and promote endosomal escape. GFP assay was used to evaluate the effect of target gene silencing in vitro. The results suggest that LNP with DOPPPZ or DHSMPPZ may have the same level of target gene silencing effect as cationic lipid produced by other company. The blood retention was confirmed using mice. The results showed that DHSMPPZ significantly improved blood retention compared to DOPPPZ. The results of measuring the concentration of ionized lipid in the blood are shown here, and it was confirmed that DHSMPPZ present in the blood in greater amount than DOPPPZ. The half-life of siRNA was also checked using a fluorescent plate reader, and it was found to be about 6 times longer than the DOPPPZ. These results suggest that possibility of extrahepatic targeting such as cancer. This is summary. We succeeded in synthesizing charge reversible lipid having pH responsiveness by one-step enzymatic reaction. Charge reversible lipid was able to encapsulate siRNA and change their charge according to pH. LNPs composed of charge reversible lipid had low cytotoxicity as compared with LNPs composed of cationic lipid. LNP composed of charge reversible lipid show effective RNA interference. It was suggested that LNPs composed of DOPDEDA or DHSMDEDA deliver siRNA to the liver. DHSMPPZ may improve the retention time of LNPs in plasma. This is all for my presentation. Thank you for your kind attention.
谢谢 Mr. 于龙伟的介绍。那接下来是 Q&A 的时间。那在场或是线上的嘉宾有没有什么问题想要请教 Mr. 于龙伟？Mr. Uday, I have a question for you. Can any drug be encapsulated using your Prisome technology?
Okay. Prisome can prepare liposome by simply adding a chaos solution and stirring. We consider it can be easily changed from other liposome preparation methods.
So I have a follow-up question. So if I have a liposome that has been manufactured with other preparation method, what would it take to be like transfer into like your fabrication technology?
Liposome technology can easily to prepare liposome and exhibition encapsulated liposome made almost same with docile ones.
OK, thank you. 不知道在场或者是线上的嘉宾还有没有其他的问题？如果没有的话，Mr. Wu, I have another question for you about the lipid nanoparticle for nuclei delivery. I saw you have using your ionizable your lipid for siRNA like delivery. Have you tried to use your technology for mRNA delivery?
We try to messenger RNA delivery, but messenger RNA LNP is messenger RNA delivery we try now, so we can't show the result now.
Okay, so we will look forward for your like future work when applying your technology to mRNA. 好，那如果在座没有任何其他的问题的话，让我们再一次掌声，谢谢 Mr. Inoue。那今天大会第二位请到日本的讲者是 Dr. Tasuhiro Ishida。石田教授是德岛大学医学牙科药学研究科的教授，目前也兼任产学院研究开发事业部门的教授。石田教授发表过超过200篇的论文，并且他在微肢体的研究发表多次获得了日本药学会年度 Highly Cited Review Award。那这一次 Dr. Tasuhiro 要跟我们分享的是在微肢体当中一个非常重要的成分，也就是PEG 的 lipid， 它常常会有 anti-PEG 的 immunity。那所以 Dr. Tasuhiro 今天要跟我们演讲分享的就是 anti-PEG immunity, the impact on the development on pegylated products。让我们掌声欢迎石田教授。
I am Dr. Ishida, who is a professor, Department of Pharmacokinetics and Biopharmaceutics, Tokushima University. I am so sorry I could not visit Taiwan this time due to COVID-19 pandemic. I hope I could come to Taiwan in the future to see you directly. First of all, I would like to thank organizing committee members for giving me this great opportunity to give a presentation today. The title of my talk here, Anti-PEG Immunity, the Impact on the Development of PEG-related Products. In this slide, I just show up the outline of this talk. First, I will briefly introduce pegylation and its impact on the modified materials. And then I will explain the ABC phenomenon and the mechanism behind the phenomenon. Then I will touch pre-existing anti-PEG antibodies and measure causes for their induction. Additionally, I will show you COVID messenger RNA vaccines also induce anti-PEG immunity. Finally, I will summarize what I showed you briefly. So let's move to main topic. As you know, pegylation is still gold standard to develop particular drug delivery systems and protein drugs, so on. It's believed that pegylation increases biocompatibility and retention time of nanoparticles and proteins, and reduces immunogenicity of nanoparticles and proteins. Pegylation gives a liposome a long circulating property in blood, leading to a passive targeting of pegylated liposome due to enhanced permeability and retention effect, EPR effect. As you know, active solid tumor and inflammatory tissues have blood vessels with high permeability. So long circulating liposomes with 100 nanometer are passively accumulated. These pictures help you to understand how pegylated liposome extravasate from leaky vessels. We induced new blood vessels under the mouse skin artificially and injected fluorescence labeled 100 nanometer pegylated liposome intravenously. As you can see, fluorescence, which means the liposomes, mainly accumulated around invisible tiny blood vessels, but not around the visible large and matured blood vessels. Passive tumor accumulation of long circulating pegylated liposome via the EPR effect is occurred in patient. This slide is from homepage of Alza Corporation. I got this slide a long time ago. This guy unfortunately got HIV-related Kaposi sarcoma, which is a skin cancer, and received IV injection of pegylated liposome containing Ingeum. As you can see, the liposome were accumulated in tumor regions in head, shoulder, and left legs. In a time after injection-dependent manner. On the basis of such characteristics, pegylated liposomal doxorubicin formulation, namely Doxil or Carex, has been approved for clinical use in 1995 in the USA. So people believe pegylation results in many advantages. However, in 2000, a Netherlands group reported strange phenomenon upon repeated injections of pegylate liposome. They seem to develop radio-labeled pegylate liposomes for tumor diagnosis. They seem to study PK of each dose pegylate liposome for sure, with certain intervals. However, they found out the second dose was rapidly cleared from blood circulation and extensively accumulated in the liver. This result indicated pegylate liposome may have immunogenicity. This is actually the first report regarding the ABC phenomenon upon pegylated liposomes. Since then, we continuously investigated the mechanism behind the ABC phenomenon to pegylated liposome and pegylated liposome. Both pegylated liposomes and pegylated proteins induce the ABC phenomenon via production of anti-PEG IgM and IgGs. But those have different mechanisms to induce anti-PEG antibodies. In this slide, I just focus on pegylated liposome, so please do not make you confusion. Briefly, first dose 100 nanometer pegylated liposomes reach spleen due to their long circulation property. They meet marginal zone MZ B cells expressing anti-PEG IgM on their surface and stimulate the cells for 3 to 5 days without T cell help. The B cells begin to secrete anti-PEG IgM into blood circulation. The production reaches peak level at day 5 to day 10. Anti-PEG IgM exists for 2 to 3 weeks due to its biological half-life. If anti-PEG IgM was in the blood circulation, the second dose pegylated liposome were immediately recognized by the IgM and formed immunocomplexes. IgM is not eat-me signal, so the immune complex with IgM needs to activate complement system to clear them. So the immune complex composed of pegylated liposome, anti-PEG IgM, and several complement components are rapidly cleared from blood circulation via complement receptor-mediated endocytosis by Kupffer cells like this. At the same time, complement activation releases anaphylaxis, such as C3A and C5A. The immune complexes, including complement components, may stimulate allergy mediator secretory cells. So, anti-PEG antibody may affect efficacy of subsequent dose pegylated materials as well as become one of major causes of fatal anaphylaxis to pegylated materials. We are currently very interested in pre-existing anti-PEG antibodies in humans. Last decade, several papers reported a large number of people who have never received pegylated drug formulation has anti-PEG antibodies. This is our own result. We obtained 200 donated sera of volunteers through Japan Red Cross. We detected anti-PEG IgM, but not IgG in our case. But anyway, we could confirm there exist people who have pre-existing anti-PEG IgM in Japan. Several people has a quite high level of anti-PEG IgM. This study was carried out more than 4 years ago. Recently, we received messenger RNA vaccine composed of pegylated lipid nanoparticles. So we assume the percentage of the high level of anti-PEG IgM more increasing recently. We wonder if PEG in the cosmetics or medicines is a major cause of inducing anti-PEG IgM in human who has never received PEG medicines. You may be shocked, but PEG is everywhere around us. So too difficult to avoid exposure to PEG in daily life. We tested the issue of whether cosmetic induces anti-PEG IgM. We put cosmetic daily for 7 days and 14 days and 12 days to mouse with or without dermal roller treatment. Dermal roller has large number of short needles here. So the treatment with dermal roller make many small holes on skin, which increase penetration of component of cosmetics. As you can see in the graph, in normal mice, intact skin, anti-PEG IgM was detected at day 14. In dermal roller treated mice, compromised skin, anti-PEG IgM was detected at day 7. These results suggest that cosmetics might be one of major causes of inducing anti-PEG IgM in healthy people. So you may think what happens if anti-PEG IgM positive people receive COVID-19 messenger RNA vaccine composed of pegylated lipid nanoparticles. We will give you the answer here. We induced anti-PEG IgM by cosmetics as shown in last slide and injected Pfizer's messenger RNA vaccine mimicked formulation intramuscularly here. We labeled messenger RNA LNP, which fluorescence to see distribution of LNP, and encapsulated luciferase messenger RNA to visualize protein expression. As you can see here, dial distribution are almost same in each group. The protein expression at injected region here. Also same, regardless of presence or absence of unspec IgM. So we could say pre-existing unspec IgM does not affect efficacy of COVID-19 messenger RNA vaccine. But additionally, you might focus on here, stomach region. In control mice, Luciferase activity was detected at not only leg here, but also stomach region. In unspec IgM positive mice, no activity in stomach region. We assume unspec IgM brought messenger RNA LNP absorbed into the blood circulation after intramuscular injection into Kupffer cells to digest, not hepatocytes to messenger RNA expression in liver. As I explained, removal of pegylated materials by unspec IgM needs complement activation. So we wonder, messenger RNA LNP vaccination to people who has unspec IgM might induce anaphylaxis reaction too, as you read in newspapers or something. We then tested effect of unspec IgM induced by cosmetics on therapeutic effect of pegylated liposomal formulation. In this case, we choose Doxil to test. We began 5 days before tumor inoculation, the topical application of cosmetics together with dermal roller treatment to induce unspec IgM for 7 days. Doxil was intravenously injected at day 5 and 8 with such doses. After tumor inoculation here. As you can see in the figures here, in the no-unspec IgM animal control, Doxil. Doxil treatment strongly suppresses tumor growth. On the other hand, in the unspec IgM positive mice, cosmetics, the tumor growth suppression effect of Doxil was slightly suppressed. This result indicates pre-existing unspec IgM may affect efficacy of pegylated medicine intravenously administered in some cases. You may have another question. Does messenger RNA vaccine induce unspec IgM because Pfizer's and Moderna's are both composed of pegylated LNP? So we injected pegylated messenger RNA LNP intramuscularly or intravenously. The schedule is here. We injected messenger RNA LNP twice with 3 weeks intervals. And then we collected serum from the mice as indicated time points. As you can see here, vaccination induces anti-PEG IgM regardless of injected route. So unfortunately, large number of people who received messenger RNA vaccine might become anti-PEG antibodies carrier. So we should pay further attention to this issue. Otherwise, many patients may lose large amount of money without expected therapeutic efficacy of PEG-related therapeutics, because the therapeutic might immediately eliminate from blood circulation. This is a summary of my talk. PEGylation is still gold standard to develop formulations with proteins, nucleic acid, and nanoparticles. PEGylation may increase immunogenicity of PEG, resulting in inducing anti-PEG antibodies. The ABC phenomenon, namely induction of anti-PEG antibodies and rapid clearance of subsequent dose, is problematic. Pre-existing anti-PEG antibodies induced by daily use materials such as cosmetics and, unfortunately, COVID-19 messenger RNA vaccinations recently might decrease patient QOL by decreasing therapeutic effect of intravenously injected PEG-related drug formulations and by leading severe anaphylaxis in some cases. However, it seems that anti-PEG antibodies do not necessarily induce the ABC phenomenon to all PEG-related drug formulations. Probably, they are polyclonal antibodies having different ability and affinity to PEG or even PEG-related materials. We are continuously researching these issues in our laboratory. This is take-home message. We need to live in the world existing a large number of people having pre-existing anti-PEG antibodies. So we need to continue research and study and investigate these issues forever. This is the final slide. I really appreciate all members of my laboratory, their effort and big patience. Also, I would like to thank Foundation and our government for research grants. That's all. Thank you very much for your kind attention. Good afternoon, everybody. I am Dr. Ishida, who is a professor, department
a talk. So 我们现在就是接受现场或者是线上的来宾有没有问题？Dr. Tatsushiro, we have a question from Taiwan. Since people expose at different PEG-related products so often, why there is no anti-PEG IgG detected in serum?
You mean the IgG?
Yes, you mentioned the IgM, the anti-PEG IgM in serum, but since we are exposed to like so many PEG products, why don't you detect any IgG in human body?
Yes, that's a good question. The several papers says, the normal human has anti-PEG IgG, but in my laboratory, we have never detected such IgG to PEG. The reason is still unclear, but polyethylene glycol PEG is generally the antigen, which is T cell independent antigen, they say. So, which means the polyethylene glycol would not reach the memory of immunological response. That is the answer. But only one possibility to induce the anti-PEG IgG is the high immunogenicity protein is conjugated to PEG. For example, over-albumin. In that case, PEG-OVA sometimes induce anti-PEG IgG, but not so often.
Thank you. So we have another question. To your knowledge, is there any clinical report showing patients receive Docile treatment, develop antibody against Docile, and losing treatment efficacy?
We have studied such issue with the clinician in Tokyo University. We detected actually some patients with the breast cancer, high anti-PEG IgM titer. Then we was anxious about the ABC phenomenon in such patient. But when we injected the Doxil, nothing happened in terms of the PK of Doxil as well as the anaphylaxis. So we concluded Doxil has a high lipid dose. So it means that tons of particles in there. So, some anti-PEG IgM bound to the Doxil particle, but so many particles there. So, some particles should be gone to the liver Kupffer cells, but another part of the particles can reach to the tumor region. So it works. So we need to pay attention to the dose of the second dose. If we inject tiny numbers of vesicles or tiny amount of proteins, it's easily gone. But if we injected tons of proteins, tons of vesicles, it's no problem. Okay?
Okay, thank you. 在场有没有其他的贵宾有问题的？如果没有的话，那我们再一次掌声谢谢 Dr. Tashihino。Thank you。那接下来是中场 break 的时间。那我们在会场的后方有提供就是茶点跟饮料。那请各位贵宾在3:20的时候回到座位。那我们继续下半场的分享，谢谢。 台湾就是微肢体相关的研究。那首先我们邀请到的是工研院生医所郑平福博士。那郑平博士毕业于国立台湾大学生化科学系，那是在工研院有多年药物传输跟新颖载体开发的经验。那今天他要来跟我们分享的题目是 The Trend of Lipid Nanoparticle Technology for Oligonucleotide。那我们掌声欢迎郑平福博士，谢谢。
Gentlemen, it's my honor to report the research results of mRNA in E3. The title of my speech is The Trend of Lipid Nanoparticle Technology for Oligonucleotides. I'm Pingfu Chen from Taiwan E3. The first is my personal background. My PhD is from NTU Biochemistry, and my work experience is all in E3. The mRNA LNP team is about 25 people, and my role is to lead the lipid design and the synthesis teams. The LNP is 5 components, mRNA, and 4 different ratios of lipids. And for me, the LNP pictures look like the colorful hot air balloon. The outline of today is the trend of the oligonucleotide drugs, key technology for mRNA LNP, development results of mRNA LNP in E3, and the summary. As shown in the figure, oligonucleotide drugs are most short-chain nucleic acid before 2020, and about 9 drugs have been approved. In these 20 years, the development of nucleic acid drugs has focused on chemical modification and targeted delivery. After 2020, due to the COVID-19 pandemic, the success of long-chain mRNA vaccine has resulted in the rapid development of mRNA LNP technology. Now to see fast-growing mRNA markets, COVID-19 vaccine prove feasibility of mRNA products. The sales revenue of mRNA products are up to 23 billion in 2035. The sales of COVID-19 vaccine will decrease years by years, but vaccine related to infectious disease will still be the main one. The next therapeutic potential of mRNA is expected to be infectious disease, cancer vaccine, immuno-oncology, genetic disorder therapy. Delivery technology is critical for RNA products because delivery is very important for RNA stability and the rate of the endocytosis. According to pattern analysis result, in addition to RNA design like self-amplified, cap, poly-A, sequence modification, delivery technology is also rapid growing up in these 10 years. Next, let's look at the pattern network analysis of COVID-19 mRNA vaccines. As shown in the figure, we can see the relationship with each other is very complex, from Moderna, BNT to CureVac. Patent legal battles against each other, from RNA design to lipid structure, LNP composition, and more. And companies such as Acuitas, Avitas, Pfizer are also involved. Next, we introduce LNP delivery technology. The main function of LNP are protect nucleic acid and release them into cells. There are 4 lipids in LNP: helper lipid, auxiliary lipid, cholesterol, PG lipid. Compared to virus, the advantage of mRNA-LNP are no genome integration, low oncogenic risk, high loading capacity, no pre-existing immunity, low cost. LNP play a major role in endosomal escape. First, LNP are neutral charge to avoid toxicity. After endocytosis, in the acid condition of the endosome, it is positive charge. Then interaction with the endosome membrane and escape from the endosome, release the mRNA into the cytosol. As mentioned, RNA are key components of LNP. This is RNA structure. For the head group, it could be secondary, tertiary, quaternary amine, which are positive charge under different conditions. There are also some heterocyclic are used in this part. Next is the linker. The functional group in this part could be ester bond, amide, carbamate, disulfide bond. Finally, the design of the lipid tail could be saturated, unsaturated, or complex structures, and usually branch, unsaturated, and the complex structure have better effect. As shown in these figures, the RNA structure and the composition ratio of LNP is the core of patent. From the structure of 3 approved products, MC3 lipid, SM102, LC0315 lipid, there are some similarity could be pointed out, like tetraamine, OH group, and the biodegradable ester bond. Then 50 to 65 molar ratio of lipid in the LNP is patented by Abutas. Next, to see the result of each mRNA LNP, here we report 2 cases. First, in vivo mRNA delivered to liver. The second is ex vivo transfection of immune cells. Development of mRNA LNP for in vivo and ex vivo applications. First, in vivo, delivery of IL-21 mRNA for the treatment of HBV infection. And for the ex vivo, the goal is to transfection immune cell, such as CAR-T, CAR-NK, with high transfection efficiency and controllable expression. Then we see the IL-21 mRNA LNP for HBV. Using a microfluidic system to establish continuous LNP formulation preparation. The preparation is highly consistent in our results. As shown in the picture of the results, you can see 10 batch preparation. The size average is 100 nanometer, PDI below than 0.1, and the EE is about 90%. And this particle size. For the LNP preparation, first we set up TNS PK assay by DOP-DEDA LNP. The DOP-DEDA lipid is provided by Zedong fine chemicals. The structure is shown here, and we use different ratio of lipid in the LNP. The titration result of 4 groups, you can see from 5, 6, 7, 8. The PK is 6.4, 0.5, 0.6, 0.7, and they are within the range of the 6 to 7. Now to see the innovate identifiable lipid for mRNA LNP application. Our development strategy is to screen different functional groups that enhance nucleotide affinities. The screen protocol is a new lipid design and synthesis, identification and quality analysis by LC-MS MMR, then LNP formulation. After screen, we find 4 lipids with good transfection efficiencies. First is the P series, the P2, and the P6 lipid. Their pK is 6.9, and the purity is analyzed by the HPLC ELST. This shows the results. And the other series is 2 T lipid series, T2, T4. The TNS pK is 6.6 and 6.3, and also the purity analyzed by the HPLC ELST. Now to see the in vitro transfection efficiency of novel lipids. We use the cell to express the dosimeters, and this is the expression levels, and to compare the different lipids. This is the cell only, and SM102 LNP, MC3 LNP. This two is our reference standard to compare our lipid. We have 4 types lipid: P lipid, R lipid, and S lipid, T lipid, with different functional group in the head of the lipid. And this result shows P2, P6, and the T2, T4 has the good transfection effect, which are better than MC3 or similar. And these 4 lipids are following test the IL-21 mRNA transfection. Then to see the in vitro transfection efficiency of human IL-21 mRNA, the result is shown in the figures. The y-axis is the IL-21 concentrations, and the S is the different time of the cell expression. The gray one is the cell only. Green one is SM102 LNP. The red one is MC3 LNP, and the blue one is our lipids. You can see from these pictures, our lipid with good transfection efficiency, better than the MC3, and near to the SM102. And this is the standard curve. So the immunopD study is ongoing, and we still developing the lipid. Hope that in the future we can have the lipid that can better than SM102. Next, let me move to the ex vivo part. This part is the immune cell transfections, and main target is the NK cells. Since the NK cell has more possibility from multiple different cells, and not limited to the only patient self, and to be expect have more application in the future. Let me introduce immune cell therapies. In 2017, the first CAR T therapy approved, and bringing the immunotherapy market, which is considered a medical breakthrough. More recently, market has been focused on other types of immune cells, such as CAR macrophage and CAR NKs. And at the same time, cell therapy is also tried to apply into the area other than the cancers, like infection disease, diabetes, autoimmune, and fibrosis. Next, to see the comparison of the delivery technology for immune cells. The main transfection tools for immunotransfection is the virus transduction, electroporation, nanoparticles. Compared to these two technologies, the LNP is easier and inexpensive, and low cytotoxicity and low immunogenicity. For the application, the LNP is more potential for offshore therapies. The major player in this area is the precision nanosystems in sonography. Then to see the each LNP for immune cell transfection. Each LNP has high transfection efficiency, about 75%, as well as good cell viability on NK. You can see from this data, the y-axis is the percent of the cell transfections, and then the other one is the protein expression levels. You can see each LNP has the same result to the SM102, and this two is better than the MC3 and the lipofetamine. And this data can also confirm from the fluorescence microscopy. You can see the SM102 and the HLMP are much better than the MC3. And now the prime NK study is ongoing. Then to see the comparison of each LNP transfection with other published technologies. In cell lines, delivery efficiency of each LNP is super than retrovirus, similar to the lentivirus and the electroporations. For the cell variability, each LNP has super than electroporations and the CAR polymers. You can see from these tables, our efficiency is about 75% compared to other technology efficiency, the retrovirus and the lentivirus and the electroporations. But now we are the prime NK study is ongoing, and the cell variability is better than the electroporations. Finally is the summary of N9 LNP result in E3. We are dedicated in developing next generation RNA LNP technology for ex vivo and in vivo therapies. And now there are 4 novel lipids P2, P6, T2, T4 with good transfection efficiencies which are better than MC3 or similar. And this is our mRNA LNP process in E3 from the formulation and the microfluidic purification, filtration, QC assays. And we welcome any collaboration opportunities. We're looking for companies interested in commercialize our valuable lipid and the development product with our novel LNP, and also looking for collaboration opportunity with biotechnology or institute who is developing circular RNA and the iPSC derived NK cells. This is our contact email. Welcome. The final one is an acknowledgement. First, thank you Zilong Fine Chemical to invite me, and this project is supported by Taiwan MOEA, and this our E3LNP teams. The project leader is Felix Chen, and the ADP team, and the formulation team, and the functional assay team. This is our talk. Thank you.
让我们掌声感谢郑平福博士的。谢谢郑平福博士的演讲。那我们现场开放，就是有问题想要询问郑平福博士的。 好，那这边线上有个问题是在投影片的16页，就是当你改变不同的lipid composition 的时候，你的 DOP、DEDA 的 pKa 是会怎么样被改变吗？
Thank you for the question. And because we use two lipid in this LNP, so we use DOP, DEDA, and our new carbon lipids. The two lipids will the pKa is contributed from two lipids, so the pKa will change. So you can see the different composition the LNP. So the different pKa because the different ratio of the two carbon lipids.
好，谢谢。那如果说就是对 LNP 传输核酸不是那么了解的，就是这个领域没有这么多深入了解的领域，就这部分你可以稍微解释一下，就是我们所谓材料的 pKa 跟 LNP 的 TNS 的 pKa 它的差别，跟在您研究开发核酸的传输上面，它有什么重要的指标意义？
所以我是直接用中文回答。
我是用中文讲。
OK， 那应该说就是材料本身的 pKa 以 chemical 预测出来 chem 做预测出来，大概会是比就是 LNP 预测出来，因为 LNP 的组成其实还有4种 lipid， 所以它的 pKa 是综合的一个滴定出来的结果。那以材料本身 carbon lipid 的一个 pKa 大概chem 就画出来会是比LNP 低出来，大概会高2左右。以平均值来讲，我们统计过，已经大概 publish 这些lipid 的一些pKa 的情况，大概都是比LNP 低出来高2到3左右，所以我们是在这个范围里面去设计不同的一些 lipid 的结构，让它可以就是落在比如说8到9点多的一个范围里面，然后让它组合出来的一个 LNP 的 pKa 才会落在6到7的范围里面。
好，那现场的来宾有没有其他的问题想要请教郑平福博士？
像你们把譬如说GLP 的 mRNA 或者是 luciferase 包在 LNP， 有没有去看过它的 release 的 profile 嘛？就说包的因为包是一回事嘛，释放才是重要的。
对，
有没有看过一些 release 的一些 study 吗？
我们在 cell level 看过 ，cell level 基本上会看得到说表现的一个情形，因为你刚刚有看到它就是在不管在protein量或者是在哪些细胞被传染的一个情况下，会有一个表现的值。那本身的时候其实也打入到活体的部分去看到。那以这个 case 来讲的话，我们主要的还是会到 liver 为主，然后再来是 spleen， 这是以活体小组看得到的一个表现亮度来讲。对。
那我补充一下，因为我本身有参与这个计划。就是我们在做，一般我们 liposome 如果包小分子的话，小分子要离开 liposome 才会有药效，所以我们通常都会做 in vitro 或是 in vivo 的 release。但是以核酸来说的话，其实它出现并不代表它有效果。siRNA 它必须要真的把 RNA silence 掉 ，mRNA 它必须要离开了之后，它必须要开始转染产生 protein。所以以核酸来说的话，其实我们通常不会做所谓的 in vitro release， 我们就会直接看就是 siRNA silencing 它的效能，或者是我们转染 mRNA 它产生的 EGFP 或者是 luciferase 的 RNA， 然后这个会是我们的指标。那如果你要看说它是有没有被吞进去，或者是它事实上是有被释放转染的话，通常我们会再额外 label 一个就是那个荧光带去看它是只有 uptake 或是 uptake 之后它有就是这一些就是 protein 的转染效力这样子。
我快速请教一个问题啊，
是
这个重要的是那个 endosome escape，
对，
那你这个 endosome escape 的 efficiency 是多少？
我们基本上我们现在的 tool 还没办法去测到 endosome escape 的一个百分比，所以我们还是以传染的一个效率来看的话，那我们会觉得说 基本上以我们的经验值来讲，它影响最大的其实是第一个是 pKa， 第二个是 lipid 的 ionized lipid 的一个外形，因为前面 publish 的 data 会告诉我们说，比较一个圆柱形的一个情况的话，它比较容易去干扰到内膜的一个稳定性，所以它比较容易去逃脱。那第三个其实是就是它核酸跟你 lipid 的 binding 的强度。那这边一个 case 来讲的话，以我们的经验值，我们一个目前转染比较好效果的一个 lipid 的话，那它本身的一个pKa 基本上一定要落在 LNP 的 pKa 一定要落在6到7，这是以我们的经验值来讲，因为超过这个基本上转染效果都不会太好。但是即使你的 LNP 的 pKa 落在6到7的时候，那我们曾经也试过比较强的一个结合力，就是说它跟核酸本身的一个 interaction 比较强的时候，那它其实包覆的效果还蛮好的，就是 98% 以上的一个包覆率。但是这个时候其实转染效果反而不好，就是说太强的结合力，即使它 pKa 落在6到7之间，但是它就是这样的一个情况下，核酸其实没办法被释放出来，所以它反而干扰到说它一个转染的效果。所以我们刚刚总结了一个3个影响 escape 的一些factor 就在那边。
好，那因为时间的关系，那我们就是掌声谢谢曾品福博士。好，那接下来进入我们今天最后的一个演讲。那这次的新冠核酸疫苗的起飞当中，我们可以知道，其实在脂质载体的制造是一门就是门槛极高的工艺，所以我们在会议的最后邀请陈金婷教授来为我们分享就是脂质载体的制造技术。那陈金婷教授是毕业于美国肯塔基大学微生物免疫学研究所，那目前是台湾大学生科系的教授。那陈教授主要的研究领域是借由药物传输系统的研发来进行肿瘤跟感染性疾病的治疗。Nano carrier using microfluidic rapid mixing， 让我们掌声欢迎陈教授的演讲。
To join this forum again. In this talk, I'm going to present some research data which could be regarded as the extended study from last year's presentation in developing liposomal drugs for cancer treatment. Last year, we showed you the advantages of using this dual-effect liposome for cancer treatment. Although the therapeutic efficacy is amazing in naturally developed tumor, to treat a 35-kilogram dog with orophabrosarcoma, we have to collect many batches of lab-scale liposomal drugs, which cost us 2 weeks to prepare for one shot. Therefore, the scale-up feasibility for manufacturing this dual-effect liposome is very critical for the following preclinical and clinical development. To solve this scale-up problem, we began to develop a microfluidic rapid mixing approach for fabricating these dual-effect liposomes. Before the data are presented, let me provide some background information which you might be familiar with. Lipid-based nanocarriers, in fact, include liposomes, solid lipid nanoparticles, nanostructural lipid carriers, and lipid nanoparticles for delivering small molecular drugs as well as biologicals. These nanocarriers can effectively deliver active pharmaceutical ingredients, the API, with the advantages of altering the pharmacokinetic properties, reducing the side effect and increasing the bioavailability. Liposome and lipid nanoparticle, the LNP, are the two most well-known lipid nanocarriers. They are similar but different drug delivery nanocarriers. Liposomes are considered as nano-sized colloidal vascular structures composed of an aqueous core surrounded by one or more phospholipid bilayers. Doxia, the most famous liposomal drugs used in clinical, was prepared by encapsulating doxorubicin in the aqueous cold using active remote loading via ammonia sulfate gradient. Lipid nanoparticle, the well-known RNA delivery vehicle, such as Moderna or BNT COVID-19 vaccines, are created by mixing a specific combination of phospholipids, ionizable lipids, cholesterol, and pegylated phospholipids. In fact, LNP are regarded as solid particles, encapsulate drug molecule in the non-aqueous cold, assuming a reverse micelle-like structure. The current fabricating method for liposome is a top-down approach using monolipid hydration and high shielding mixing, such as homogenization, sonication, and freeze-thaw method. Initially, plain bilayer structures are formed. Via the input of high energy, these larger lipid bilayers are further broken down into small liposomes, which could be further downsized by membrane filters. However, in developing liposomal drug for clinical purpose, these conventional top-down methods still face many challenges in the manufacturing process, such as the poor scale-up feasibility and large batch-to-batch variability due to the lack of control over experimental variables. Recently, a bottom-up approach, microfluidic rapid mixing, takes a much different approach. These methods build lipid nanocarriers by mixing the organic stream containing lipid components and aqueous stream in a microfluidic channel by nominal mixing. Most of the variables could be precise controlled in this approach, such as the organic solvent, lipid concentrations, aqueous buffer, and total flow rate and flow rate ratio used in the mixing process. The mixing process is finished within seconds, and nanoparticles are formed by self-assembled mechanism. Lipid nanocarriers prepared by microfluidic technology are more controllable and reproducible during the manufacturing process. In fact, microfluidics not only allows the generation of small quantities of nanocarriers for the initial screening experiment, but also for large-scale quantities for the following preclinical and clinical translational development. The parameters used in small-scale preparation could be applied to larger scale. In other words, the scale-up process is linear. Except for the microfluidics rapid mixing, we also implement quality by design, the QPD approach in developing lipid-based nanocarriers. So what is QPD? In pharmaceutics, QPD is a systemic approach for formulation development that begins with the predefined objectives and emphasizes the product and process understanding and processing control based on sound science and quality risk management. These mathematical and statistical methodologies allows for developing efficient pharmaceutical formulation and control methods. So how do we get started in using this microfluidics approach? The first step we start in using this microfluidic approach is to find out the preformulation of lipid nanocarriers, which could be used to load positively charged hydrophilic drugs. Initially, doxycycline was used as a model drug due to its high solubility and ionized property in aqueous environment. I will not go through the experimental details for this preformulation development in microfluidic setup. Briefly, through the QPD approach, we obtain the critical parameters such as the composition and relative ratio of lipids and factors in microfluidic process during the development, aiming to have a stable for pre-formulation of lipid nanocarriers. And as you can find here, the pre-formulation we found is composed of EPG, HSPC, cholesterol, DSPE, and PEG 2000. Egg-phosphatidylglycerol EPG used in the present formulation was to increase the association between doxycycline and the carriers. And the main content in this presentation is to verify whether this preformulation could be used to encapsulate the chemo drug doxorubicin. Formulation optimization for these dox-loaded lipid nanocarriers, the LNC dox, was further developed based on the QPD approach. In the end, I will further show you the structural and therapeutic efficacy of the optimized dual-effect liposome loaded with doxorubicin and fluorescent synthetizer CE6 by the microfluidic technology. So as mentioned above, EPG was used in this formulation to increase the association between doxorubicin and carriers. As you can find in the cryo-ETM images, the morphological structure of dox-loaded nanocarriers prepared with or without EPG is quite different. Nanocarriers without EPG exhibited the traditional bilayer vehicles of liposomes. However, in the presence of EPG, complex of doxorubicin and lipid were found in the structural code of LNC dox, which was quite similar to the RNA-loaded lipid nanocarriers prepared by microfluidics, and the traditional liposomal dox prepared by the traditional method contains dox sulfate crystal in the middle. Dox-loaded nanocarriers without EPG in fact show low drug-to-lipid ratio compared to the LNC dox containing EPG. There is about 3.54 difference. In fact, the drug-to-lipid ratio of these LNC dox developed in this study is also significantly higher than that of a published report using anionic lipids or liposomal dox prepared by this traditional method. The electrostatic interaction between cation, doxorubicin, and anionic EPG could be the main force for the increased drug encapsulation during LNC formation process. This slide shows you the proposed mechanism of LNC dox formation by microfluidics. During the process of microfluidic lipid mixing, the electrostatic interaction between the anionic EPG and positive charged doxorubicin form inverse structure. These interactions further act as nucleating agent and finally form packed structural code in the lipid nanocarriers. This explains the electron-dense code found in the cryo-EM imaging of DOX-loaded lipid nanocarriers. So how about the characteristics of these LNC DOX? As shown in the left panel, the accumulated doxorubicin release from LNC DOX was about 32.5% during the first 24 hours at neutral pH. However, DOX released from liposomal DOX prepared by the traditional ammonia sulfate gradient was about 19.2%. In the acidic environment, the accumulated drug release from LNC DOX was about 54.7% compared to the 12.5% of lipodox. This result suggests the preferred release of LNC DOX in an acidic tumor microenvironment. 64.2% and 80.8% of the dogs remained inside the LNC dogs and traditional liposomal dogs after 24-hour incubation with serum protein, suggesting the serum stability of LNC dogs was lower compared to the liposomal dogs prepared by the traditional method. To evaluate the anti-tumor effect of LNC dogs, an in vivo therapeutic study was performed using 26 syngeneic biopsy tumor model, as shown here. The tumors became larger in the saline control and free-form dogs treated groups. However, significant tumor reduction was found in mice treated with traditional liposomal dogs and LNC dogs. Although the serum stability of LNC dogs was less stable compared to the liposomal dogs, the therapeutic efficacy showed no significant difference between these two formulations. We then further assessed whether the improved serum stability of these LNC dogs can significantly enhance the therapeutic efficacy. As mentioned above, DOX-loaded lipid nanoparticles exhibit an electron-dense core forming from the electrostatic interaction between the EPG and cationic DOX. In this regard, one approach to increase the stability of LNC DOX was to further increase the stability of internal structure of this drug carrier. Critical packing parameter, the CPP, is a theoretical number that could be used to determine the type of aggregation formed by amphiphila. It has been reported that lipids with high CPP value might facilitate the co-formation of nanocarriers, leading to high stability. Therefore, we argue that the stability improvement of LNC dogs could be improved by adjusting the lipid composition based on the phospholipid CPP. For the optimization study of LNC dogs, anionic phospholipid DOPA and TOPG was used to substitute the EPG, while the neutral phospholipid HSPC was replaced by DOPE and DSPE to examine whether the serum stability could be improved. As shown in the right panel, replacement of EPG with DOPA, the accumulated drug release was reduced to approximately 24.3% during 24 hours indication the increase in the LNC tox stability. Furthermore, substitution of HSPC with DOPE could also stabilize the LNC tox with the accumulated drug release in PBS approximately 28.1%. These results indicate that if we include DOPA and DOPE in the composition of LNC tox, the stability of LNC tox could be improved. So, with the information of including phospholipid with high CPP value, we further optimize the LNC tox loaded with doxorubicin formulation based on the QPT approach. And the best possible formulation for this LNC tox was composed of DOPA, EPG, DOPE, HSPC, cholesterol, and DSPE, PEG 2000. The particle size of these optimized nanocarriers is about 100 nanometer with high drug-to-lipid ratio of 0.23. The stability of these optimized LNC tox increased to 83.5% as compared to 66.4% of the unoptimized formulation. This optimized LNC tox remains the preferred release at acidic microenvironment. Meanwhile, the serum stability also increased to about 81%, as shown here. These results indicated that this LNC formulation could be a good nanocarrier for doxorubicin delivery with high drug-to-lipid ratio and acidic release property. So, how about the therapeutic efficacy of these optimized LNC tox? In these in vitro studies, we use commercial product Doxia for comparison. As you can find in the middle panel, there is no significant difference in the intracellular uptake between these two different dox-loaded lipid nanocarriers. However, these optimized LNC tox show higher in vitro cytotoxicity against mouse C26 tumor cell compared to the commercial Doxia. This higher cytotoxicity might relate to its better endosomal escape ability due to the property of acidic preferred release, resulting in more doxorubicin accumulated in the nucleus, as shown in the upper left panel. So these results indicated the preferred release of LNC tox in acidic environment could enhance the drug release intracellularly and enter nucleus to exert better therapeutic efficacy. In fact, these optimized LNC tox show better tumor regression and overall survival rate compared to the commercial drug Doxia in biopsy mice bearing C26 tumors, and there is no significant change in the body weight of the mice treated with LNC tox, suggesting the safety in using these LNC tox. Doxorubicin encapsulated in pegylated liposome is released both in the tumor extracellular fluid and intracellularly by endosomal escape process. However, the slow drug release and poor endosomal escape capacity of pegylated liposome prepared by the traditional method in fact impairs their therapeutic efficacy. As shown here, the present dox-loaded LNC fabricated by microfluidics has preferred drug release at acidic environment and better therapeutic efficacy in mice bearing C26 tumor. So, can this microfluidics preparation approach could be used to develop other nanocarrier loaded with different API? As you can see in this slide, in the left panel, we demonstrated the optimized dual-effect liposome with doxorubicin in the aqueous coat, which was shown as a network dox sulfate crystal and C6 in the lipid bilayer fabricated by microfluidics. Again, the complete tumor regression could be found in the mice received this dual-effect liposome fabricated by microfluidic lipid mixing. In fact, the approach we used to prepare the dual-effect liposome could facilitate the formulation development of nucleic acid loaded with nucleic acid by microfluidic technology. In our laboratory, mRNA GLP-loaded LNP formulation was developed, and the cryo-EM picture of this LNP was shown here. The expression of mRNA GLP was significant in mouse C26 tumor as found by using lipofetamine. So in this presentation, we demonstrate that strategy using the bottom-up microfluidic approach could facilitate the formulation development and optimization with the potential in improving drug loading capacity and reducing the manufacturing process for the translational development of lipid-based nanocarrier drugs. In fact, as you can find from these different cryo-EM pictures of nanocarriers loaded with different API, this method in fact can prepare nanocarriers with different morphology. For example, complexing nucleic acid during nanocarriers formation result in lipid nanoparticles. However, complexing with different small hydrophilic charged molecule might result in multiple layer liposomes, single bilayer liposomes, and lipid nanoparticle during nanocarriers formation. So this is the final of my talk, and the financial support of this study comes from the Ministry of Education and Ministry of Science and Technology Taiwan. These works are mainly carried out by outstanding student Dr. Li Jiayin and our collaborator in Formurex Pharmaceutics, Dr. Cui Min Chai and Dr. Peng Bojun provide valuable opinions in developing this lipid-based nanocarrier platform. And thank you for your listening.
谢谢陈清婷博士精彩的分享。那接下来是 Q&A 的时间，我们邀请陈清婷教授来前方。那不知道现场或是线上的嘉宾，就是有什么问题的话，可以举手让我知道。
陈教授你好，不好意思，我有3个问题想要问一下，就是有关于在第14页，你有说一个best formulation of the lipid， 那我有 curious about 说你是 try 了多少的 parameter 才会有到说你觉得它是一个 best formulation， 然后第二个问题是说，因为你说这个 microfield 的一个 mixing system， 它可以 scale out 到一个临床用药。那目前你揣测说它最大量可以到达几个 gram 或几个 kilogram 这样。那这个 system 是已经是要自己去组装吗？还是说它是 commercial 的一个 system 呢？
OK， 好，那我想我先回答你第一个问题哈，就是其实不用过去，我们到底试了多少个配方？那在今天演讲很抱歉，我对 QVT 这一件事情没有讲了很多，因为那个要讲另外1个小时。那我就简单这么讲，像在我们这个 formulation development， 你有4个不同的 lipid 组成，那你就想象，如果按照传统的方法，你这4个 lipid 组成一开始你要先猜对，你猜错了就不要玩了。你猜对之后，你要去算不同的 lipid 的组成，你每做一次，你就去做一次 assay， 每做一次做一次 assay， 你可能就要做好几百次。OK， 但是我们用 QVD 配合上这个 microfluidic system， 我们大概可以降到1/8的次数。所以这是一个，其实我今天想要跟各位分享，这是一个很不错的 approach。因为传统我们在做 formulation development， 其实最可怕的就是在试配方，每次试一个。然后改变一个条件，改变一个浓度，你就回来。那你每次看一个，可是你忘了另外还有3个，还有5个，所以那彼此间的 interaction 实际上是不容易去抓的。QPT 的 approach 事实上是一个很重要的一个概念。那它事实上也是现在美国 FDA 跟欧盟在制药产业上面开始慢慢被要求的一个 concept。OK， 那是回答你第一个问题。那第2个问题我忘记了，对不起。
想问一下，不好意思，那你 QBD 那个是你们实验室有一个统计的软体可以？
其实 QBD 事实上是一个统计，它是一个软体，是一个 commercial 软体，你可以去对不起 ，QBD 是一种 concept， 那有一些很多不同的软体可以来做这件事情 ，OK， 但是你要会设定就是说它是一个死的东西，那坦白讲你要有一点经验。我刚刚讲的，如果你一开始猜错你的主成分，那后面可能就完全不一样了。然后你做了很多实验，就是你设计几个配方之后，你可能会用一个去做实验之后回来之后，你还要去判读你的结果，然后再回来重新设计一次。所以它看起来蛮 powerful， 可是可能要累积一点经验去使用才会比较顺畅，要不然的话会在里面打转不出来这样子。那对不起，你的第2个问题。
问题是这个 microfluidic system 它最大
的问题。当初我们用 microfluidic 的系统，其实现在大家都已经听，像刚刚前面几个讲者，像郑博士也用到 precision nano system。那我有的那一套 precision nano system 是各位一定看不到，因为那是我4年前买的第1代系统。那现在已经没有那一套系统了，我也不肯跟厂商换，因为我还蛮喜欢它的。当初为什么我们会用这套系统？其实就跟一开始我所讲的，你要做一个微质体 Liposome 的 drug， 传统那种 scale up 的方式，实际上是非常痛苦的。你每一个 scale up， 你就要变一次参数，那前面可能可以用的东西，完全是可能都不能用。但是用微流体这种 bottom 的技术呢，你要 scale up， 你从一开始做2cc， 你设定的 parameter 要从2cc 变成15cc 变成1个 liter 的时候，参数的改变实际上是比较小的。你甚至要变成1个 liter、10个 liter。OK， 好，所以这是最近这几年来 microfluidic 的 system， 就是 Moderna 跟 BNT， 这些疫苗的制造能够那么快，就是靠着 microfluidic system 去制造的。那整个 scale up 当然有很多的厂商那当然大概都被绑死了，坦白讲然后就是用串联的方式去做。OK， 所以它的 scale up 是在这一边。那第3个问题是，抱歉讲到后面就忘记前面。
第3个问题就是可能刚刚那个老师有回答，就是因为你这个是已经有比较蛮 commercial 的一个设备了嘛？
有一些 commercial 的设备这个其实也可能是为了台湾的一个痛这些 commercial 的设备，有些设备呢你要买来做 research 可以，你要后来买来做 product 恐怕买不到因为它就是说。制药产业上是一个蛮可怕的产业，像刚刚我们今天提到的很多 lipid 啊什么之类的，很多时候你大家都买得到做 research 发表论文没有人会管你，可是真的要变成一个 product 会是一个很大的问题，因为你可能就不能用，因为有一些那个 patent 的问题，或者是他们商业上的一些 bundling 这样子。
陈教授您好，不好意思，想请教一下，就是您最后有提到说，这样子的Liposome， 它LNC 的 Liposome， 它包了不同的 API， 可能有不同的 morphology。那我想请教的是说，主要会影响这些 morphology 的主因是像是 API 嘛，或者 lipid 组成，或者是这些 process 的参数会影响。那另外是说，这种不同的 morphology 对于这些药物的 release 会有什么样的 impact？ 谢谢。
这是一个蛮好的问题哈。我们该这么讲，你用 microfluidic， 因为其实我们去想象我们在做 lipid nanoparticle， 它就是一个 mixing 嘛，它就是混合。传统的方法为什么不好？因为那个混合没有办法控制嘛，所以为什么有 microfluidic 的 system 可以来精确的控制它？好，所以各位可以去想象，在这个 mixing 的过程当中，你even是相同的 component， 你有不同的组成，然后你在 mixing 参数上的设定如果不一样 ，mix 的快一点 ，mix 的慢一点，你可能就会出现不同的组成。坦白讲，会，我们的经验是会。那这个有赖于后面的后置，你要怎么知道去改变条件，让它能够比较 homogeneous？ 所以很多的 release 的 property 又跟这个事实上是相关的。这个问题其实就是说，各位如果还记得刚刚你碰 Fine Chemical 他们在呈现他们 LNP 的一些GLP 或 knockdown 的时候，各位可以发现，其实那 knockdown 的效率大概都不到 50%。我们在做分身的时候，如果用 lipovitamine 送一个 siRNA 进去，那当然只有 30%， 你大概会把厂商叫来说，你这个设计的是一个很烂的 siRNA。好，那其实 release 是一个，就是说我们要看很多 GLP 或 luciferase， 我们后来是发现，其实它有一点会麻醉我们，因为你只要有一点点有 GLP 的荧光，看不清楚，光再调强一点就看得到。那这个我觉得会是一个问题，这个其实就是跟这些在 mixing 的过程中，这些形成 lipid nano 的 carrier， 我喜欢用 carrier 是因为它包括很多种。那你的形状不一样，你的组成不一样的时候，你释放出来的，我相信在小分子会有这个 issue， 在大分子大概都会有这个影响。所以你怎么去调那个组成，做出你要的东西，那就要回到你最基本的点。你希望你的 product， 你的东西是一个什么样子？要从这边回来设计你要的组成跟甚至它的形状这样子。
好，谢谢陈教授。因为我们在等一下还有一个就是所有的讲者的 panel 的 discussion， 那所以我们陈教授的 Q&A 就先到这里结束。那
谢谢。
在场的贵宾如果还有问题的话，等一下我们可以在 panel discussion 的时候来讨论。那我们再一次掌声，谢谢陈教授。好，那谢谢Mr. Yuki Inoue、Dr. Tashino 跟郑平博士还有陈廷宙教授的分享。那我们这一次台湾国际微肢体的研讨会 cover 了几个很重要的议题，一个是在微肢体的重要的脂质成分，在不同的新应用的时候，它有一些不同的新的材料设计的需求。那我们也 cover 到了在微肢体的制造上面，就是它是一门门槛很高的工艺，所以使用现在在制药产业的 QBD 其实是一个很重要的趋势。那用这样子的方式的话，来去确保我们整个 quality 的整个产品的品质，那这个是一个在药物产品一个非常重要的趋势。那我们也讲到了，就是因为这一次的其实已经存在很久的用流道的制造技术，那其实是因为这一次的 mRNA 的疫苗，其实它变成了一个显学。那我们也在这一次就是 cover 到了这样子的技术需要关键的生产的 parameter 跟一些需要注意的事项。那所以非常谢谢今天就是各位与会的参与。那接下来我想要邀请就是我们今天的讲者就是来到我们的台上，然后我们有 panel discussion。那我们先请陈金婷博士教授还有郑平福博士，然后我们的 Dr. Tashino 他会在线上也是跟我们一起来做就是 panel discussion 的部分。好，那先请线上或者是 在场的贵宾还有没有其他的就是问题想要就是请教就是今天的讲者的？好，那如果需要有人引言的话，那我先开始。Dr. Tashishiro, there is a question for you. In your opinion, how does the molecular weight and number of armor PG might influence the efficacy and side effect of the pegylated drug?
Could you give me that question again? And what's the molecular weight of PEG? You mean?
Yes, in your opinion, the molecular weight of the PEG or like the multi-arm of PEG, will it affect the drug that encapsulated in the pegylated liposome?
You mean the mouse PEG?
The molecular weight.
Yes. Of the PEG or the encapsulation of
The molecular weight of PEG, will it affect the drug encapsulated?
I see. Actually, in my experience of doxorubicin encapsulation, the molecular weight will not affect the encapsulation efficiency, but the percentage of pegylated lipid incorporated to the membrane of liposome will be strong affection. Yeah, if you put tons of the PEG, but it will be less than 15% for lipids amount. Because if you put more than 15%, the excess amount of PEG will be created by cells. But anyway, if you put a large amount of PEG, the penetration of drug will be inhibited.
Okay, thank you. So a following question, there is a like kind of a trend that's trying to discover new material to replace PEG, like the DSP PEG in like liposome because of the anti-PEG immunity. Like in your opinion, is this like a good research or like a product development like focus?
Yes. There are many approaches in the whole over the world, but unfortunately, to my knowledge, no such alternative materials are coming. But the branch type of PEG, so I mean not a single origin glycol, but like a cone structure will little bit inhibit the immunological response. But in terms of the industry point of view, the company doesn't like to do the safety assessment again with such new materials. So I think the company pays attention to avoid the anti-PEG immunity by such as a medication to suppress the immunological response.
Okay,
Did you catch my point?
Yes, thank you. So is there any other question for the panel?
Thank you for the presentation of every professor. My major is medicinal chemistry, so I'm not so familiar with Liposome. So here is my questions. Why doxorubicin is always chosen in your study? Have you ever considered other clinical drug?
Because there is a big enemy called doxia. Everybody want to replace it. So doxorubicin is always the first. This is just kidding. Because doxorubicin is easy to obtain, and the price is lower for us to do the study.
Oh.
So you can just, the result, you can compare a lot of paper to do that. So this is why we always choose doxorubicin.
Oh, thank you then.
One point I want to add for your question about doxorubicin. In fact, there are not so many liposomal drugs existing in the market, right? So the problem is because of you have to find a way to keep the chemo drug inside the liposome. Liposome is a dynamic particle. It's not rigid. So you cannot keep the drug inside the core. They will just leaking out. So I think this is why. And the other question is, the other problem, the other thing is, we scientists find a good way, like use ammonia sulfate to resuscitate it. But this method, in fact, did not apply to the other chemo drugs. So this is why most of the study we use doxorubicin.
Okay, so I have a question for Dr. Chen. So why do you choose like lipid nanoparticle for messenger RNA delivery? Have you ever considered other delivery vehicle like a polymer or like a viral vector? So what makes the lipid nanoparticle outstanding in delivery of either oligonucleotide or messenger RNA?
For my lab, we are not a synthetic lab, so we did not synthesize new lipid. And in fact, we try to use the polymer to incorporate into the LNP composition. But basically, the lipid is the base we use to develop a delivery system for the nucleic acid LNP. Just because we want to, we don't want to infringe the patent issue of the present LNP. But for the mind cells, I think to prepare the mind cell, you have to consider the CMC issue. So sometimes it's not easy. You might prepare a very good mind cell particle, but after you inject into the animal, the CMC might be breakdown, so they will quickly release. So it's not the same, and so this is why we always did the work on lipid nanoparticle. But mind cell polymer, in fact, we use is to find, just to incorporate into the composition of lipid nanoparticle.
Okay, thank you, Dr. Chen. So, Dr. Chen, do you have like any comment to add?
According to the reference result, maybe the long chain or the polymers have some issue is about like liver toxicity or the immunogenicity. So for our choice, we now use the small lipid and from the LNP And I think it's best, it have a good advantage like low immunogenicity and low toxicity. So we choose the lipid to deliver the nucleic acid drug. Thank you.
Thank you, Dr. Chen. So is there anyone have like other questions? So if not, could I ask like Dr. Chen, very similar Dr. Chen and Dr. Chen, like give us your insights at what's like the future direction or potential application of lipid nanoparticle or like liposomes.
This is a good question and big question about theoretical. I think for the liposome or LNP, I think this is the most popular one and famous one right now. For the future, I think if we want to develop the liposomal drug or LNP in Taiwan, the manufacturing process and the material, we have to avoid the patent issue. Because right now, most of the patent was holding the big pharma. So if we want to develop this liposomal drug or LNP drug, we have to think about what is the advantage we can do at Taiwan. That's what I did in my lab. We just do the work as a formulation scientist. We try to formulate, use the out of patents material to prepare the nanoparticle. And this will, in fact, can increase the therapeutic efficacy of the old drug.
Thank you.
I very agree Dr. Chen's opinion that the patent issue is the first question. So if you want to join the LNP technology, I think the patent is the first factor that you need to overcome. So we choose the lipid synthesize to overcome the patent issue. So if you want to if you want to use the LNP technology to like a vaccine or any cancer therapy, I think you have need to own your lipid. So this is we choose. Thank you.
Thank you. So Dr. Tashihiro, I have like request for you, with your expertise with pegylated product, what would be a suggestion or reminder for researchers who are going to play pegylated products?
Yeah, actually, the daily use materials like cosmetics, you guys in this morning you guys use that. Also, we received 3 or 4 times messenger RNA vaccines. So there are many people who has already anti-PEG IgM. This is a possibility. So it's true. So we just pay attention, the treatment with the pegylated materials. So, but I believe we can avoid if we know that the situation by pre-medication, So I think it's not so complicated issue. But my opinion for the liposomes or liposome future, LNP future, I just wanna head you up is the injection route. Couple years ago, no one think we inject the liposomes or LNP intramuscularly. Only intravenously, right? But now we can inject the LNP intramuscularly. So we should try to inject peritoneally, intrathecally, to the nose or lungs, even though orally. So we may be able to open the new door by changing the injection route. This is my opinion. Thank you very much.
Thank you. So if there is no further question, this concludes our session. So please give our speakers a big applause. Thank you for their talk and their sharing.好，那谢谢各位与会的贵宾。那这次大会就是圆满成功。那敬祝大家身体健康、平安顺心，谢谢。